So with that, I'd like to introduce our speaker for today. Dr. Isidoro has a pharmacy degree from the State University of Campinas. He also has a master's degree from the same university in biochemistry and human physiology. He also completed his PhD on translation, initiation, and quantitative biochemistry, at UC Davis. And since 2020, he's been part of our technical support scientist team. And today he is going to be the one providing us with an in-depth knowledge on the content that we have to share for you today. So with that, Dr. Isidoro, if you'd like to take over.
Hello, everyone, and thank you, CB, for the amazing introduction. Let me just make sure that we have everything correct, and we are ready to go. So, hello, everyone. As CB said, my name is Dr. Mario Isidoro, and I'll be conducting this webinar on the comprehensive data analysis of mRNA Seq. The focus of the webinar will be patient-derived xenografts, and it will be divided in 4 parts. So, first, I would like to talk to you a little bit about our company, Novogene. After, I will give you a brief introduction about eukaryotic RNA-seq, followed by the different data analysis that we do in our company. Lastly, I will show you a case study illustrating how we can use this tool to help us further our knowledge in biology. So, to start it off, I would like to talk to you a little bit about our company. Our network of offices and labs are global, and some of our teams are specialized. What you see on this map in blue are countries and regions where Novogene has successfully conducted projects with our clients. And the images highlight our research facilities. Especially during this pandemic time, we may need to transfer your samples to another lab in order to guarantee quality and turnaround time. Please don't hesitate to send us any questions. We are here to help. I'm also really excited to tell you that we have a new intelligent platform called Falcon. So we're adding this to help us provide even faster and more reliable data. Please notice that this provides you with a series of robots that can automate everything from sample extraction to bioinformatic analysis, reducing the TAT by 60%. So let's cut to the chase. Let's start talking about what we all came here to hear about, eukaryotic mRNA Seq. So I would like to start my introduction to mRNA sequencing by defining what is RNA. RNA is a polymer of nucleotides joined by a backbone of phosphate and ribose residues. In order to understand what is the most common function of an RNA, I have to show you the central dogma of biology. Here you have illustrated the famous central dogma of biology. It starts with DNA, which stores information inside the nucleus of every eukaryotic cell. This DNA is transcribed into RNA, which is then edited and transported to the cytoplasm of the cell, where it can be translated by the ribosome into proteins. Proteins give functionality to the cell, and as you can see, RNA bridges the information from the DNA to the production of the proteins. As I stated before, this is the most common function of an RNA, which is called messenger RNA. However, we do have other types of RNAs. So here are the most common types of RNA that we can find. First, you have the coding RNA, or mRNA, which will lead to the production of proteins, but you also have regulatory RNAs, which modulate gene regulation in most eukaryotes, and also housekeeping non-coding RNAs, like transfer RNAs or ribosomal RNAs. So now that you have a really good idea of what are the types of RNAs in the cell, their function, and how they are produced, we can move ahead and tell you a little bit more about our RNA sequencing pipeline. So here's the workflow that we use in Novogene for our mRNA sequencing projects. I won't go into a lot of detail because we have already done a webinar on mRNA sequencing, which you can find on our website. But very briefly, it starts with us receiving your sample, which is usually total RNA. After we receive your samples, we will perform our internal quality control, library preparation, library quality control, sequencing, data quality control, and bioinformatic analysis. I will briefly tell you a little bit about all these stages. So in the beginning of the project, we will receive your sample, which is usually total RNA. Our required minimum amount for our standard pipeline is 400 nanograms. Please keep in mind that we have other pipelines for low amount, low input, and ultra low input RNA sequencing. In general, we ask for at least 20 microliters of sample. Ideally, we're looking for an RNA integrity number greater than 6.8 for animals and above 6.3 for plants and fungi. We do ask that our clients achieve a concentration of 20 nanograms per microliters, if possible. This will help us ensure a more reliable outcome from the sample QC process. Here in the picture, you'll find our sample requirements, but all of them can be found in our website. Next, we will proceed to the quality control. Our primary ways of sample quality control is using the Agilion bioanalyzer system. Here at UC Davis in Sacramento, we have the Agilion 2100 system right now, and at some other labs, we have the Agilion 5400. The first thing we look at is the reconstructed gel electrophoresis in order to assess if there are any degradation. We also look at the amount of the samples, if there is any contamination, and if we have enough material to start. Once we have the results from the quality control of your samples, we will send you a report, and we will only move forward with the samples that you ask us to. Next in our workflow, we have the library construction. I won't go into extensive details, but I want to emphasize that we use trusted kits from Illumina and NAB. Shortly, we receive your total RNA. We select for the mRNA, usually using poly enrichment. Then we ensure that the pieces are the right size for the downstream processes. I want to emphasize that most of our workflow is done by robots in order to minimize human error and provide you the best data quality possible. I've mentioned Illumina a few times now, mostly because it is a sequencing platform that we favor in our company. Here, I just want to highlight a few reasons why we favor this platform. It's high throughput, it's good accuracy, it's well tested among the scientific community, and it's also very well trusted. It is for sure our choice for eukaryotic mRNA sequencing. Next, we have our data quality control. The main things we look at are first our error rate distribution, GC content distribution, which can be perturbed if our reagents are not perfect or if something goes wrong during the sequencing reaction, and the composition of the raw data. Furthermore, we have a triple data filtering system where we filter for reads containing too many uncertain base calls, we filter for too many low quality base calls, and we also filter for any read that appears to have a part of or all of the adapter. All our downstream analysis are done only with clean reads, which generally corresponds to more than 90% of the acquired reads. Lastly, we come to the BI analysis, the bioinformatic analysis. We offer several options for BI analysis, which is not a surprise, since we were founded by the biologist who wrote the SOAP and SOAP de novo, Dr. Lee. And it is a topic of the next part of our webinar, bioinformatic analysis. So on the third section of the webinar, I will talk to you about the mRNA Seq data analysis that we offer here at Novogene. As I mentioned previously, we start our BI workflow with our data quality control. We filter our data based on the error rate distribution of the reads, GC content, and also the quality of the reads. Please keep in mind that you can ask us to send you the clean reads. We can provide this triple filter reads to you as a separate data set in addition to your raw data set. Once we ensure that most of the reads in the sequencing run are clean, we move on to the next step, which depends on the fact if the genome of study has a reference or not. Here's a summarized table that our clients get with the information of our data quality control. You can see the name of the sample, the number of raw reads, followed by the number of clean reads, the number of raw bases sequenced, and the number of clean bases sequenced. Next, you have the error rate, the Q20, Q30, and GC content. Moving back to our reference genome inquiry. So if we do not have a reference genome for the sample, we can move on to our de novo RNA Seq pipeline, which will be the topic of a further webinar. Or if we do have the reference genome available, we can move forward and map the clean reads to the reference genome. From this point on, we at Novogene, we take two different paths, a structural path and a gene expression quantification path. I will start by going through the gene expression one, since it's the most common analysis asked by our clients. The first thing that we provide you with is the summary of the mapping results, which will encompass the total number of reads, the total number of mapped reads, uniquely mapped reads, multiple mapped reads, followed by the total mapping rate, and the percentage of uniquely mapped reads and multiple mapped reads, as you can see in the picture. Moving on our gene expression quantification workflow, we have our gene expression quantification. it starts with a gene expression level distribution using an FPKM distribution plot. FPKM stands for fragments per kilobase million, and it is just a normalization step for the sequencing depth and length of the genes, so that all genes in the pool can be compared and plotted together, as you also can see in the picture on the left. We will also provide you with a correlation analysis in case your study has biological replicates. So according to all gene expression level of each sample, a correlation coefficient of sample between groups is calculated, and it's drawn as heat maps. In this example, you can clearly see that the correlation between the same sample is 1, as it should be. Whereas you can also note that samples A and C are the most different with the lowest R squared of around 0.9. Lastly, we will also provide you with a Venn diagram, which shows the number of genes that are uniquely expressed within each group or samples, and the genes that are co-expressed. As you can see in this example, Group A had 780 genes that were uniquely expressed, whereas Group B had 637. Moreover, there were 11,901 genes that were co-expressed between these two groups. So, next in our workflow is the differential expression analysis and the famous volcano plot. In this graph, the horizontal axis is for the fold change of genes in different samples, whereas the vertical axis is for the statistical significance in gene expression levels. So, if you take a look at the graph, each point represents a gene. Blue dots indicates no significant difference in gene expression. Red dots indicate upregulated expression, whereas green dots indicate downregulated expressed genes. The further up on the y-axis, the higher the statistical significance. On the right, you can see in the legend the number of upregulated and downregulated genes, as well as the total number of outer genes. So the volcano plot is followed by the cluster analysis of differentially expressed genes. This graph indicates genes with similar expression patterns under various experimental conditions. By clustering genes with similar expression patterns, it is possible for you to predict unknown functions of previously characterized genes, or even to have an insight into possible functions of unknown genes. Lastly, in our gene expression quantification portion of the analysis, we have the function analysis. So the goal of this section is to find out which biological functions or pathways are statistically associated with differentially expressed genes. In this example, if you look in the figure, we can see that the cell-substrate junction is the most statistically different gene expression pattern when comparing group A with group B. This tool is particularly interesting when trying to analyze new drugs and where its function lies. So for example, by comparing a control group with a treated group, we can statistically analyze which biological pathways are changed and focus our studies on further characterize them. Now, it is time for us to move to the structural part of the BI analysis. In our structural analysis, the first thing that we take a look at is the presence of alternative splicing, which would lead to the presence of different mRNA isoforms. So every alternative splicing event corresponds to 2 isoforms, and they are represented in the figure on the left, which are referred to exon inclusion isoform, represented by an I, or exon skipping isoform, represented by an S. So if you take a look at the image on the left, assuming that we have a read that spans the end of the first black box to the beginning of the gray box, it would count as an exon inclusion isoform. Whereas if you have a read that spans throughout the region of the gray box, we would have an exon skipping isoform. And this is exactly what we show in the famous sushi plot. So on the bottom of the sushi plot, you will find the possible RNA isoforms. As you can see in this example, the top isoform indicates an exon inclusion isoform, whereas the bottom one indicates an exon exclusion isoform. In this sushi plot, you can see on the top that we had 743 reads that spammed the end of the first exon and the beginning of the second exon. We also had 677 reads that contained the end of the middle exon and the beginning of the last exon. Whereas on the bottom part, we represent the exon-skipping isoform, which had, in this case, 963 reads containing the first exon and the last exon. This plot gives us an idea of the different isoforms present in the sample, as well as their amount. So, moving on in our structure analysis, we have the SNPs, which stands for single nucleotide polymorphisms, and insertions and deletions. Here's an example of the table we provide. On the top left, we have the SNP results. As you can see, on chromosome 1 at position 14,464, we have a change from adenine to thymine. Next, we have the allele death, which represents the number of reads supporting either the reference genotype or the mutated one. So in this case, it means that we had 17 reads containing the reference genotype and 3 reads containing the altered genotype. This is followed by the genotype of the locus. We have pretty much the same structure for the table of insertions and deletions. You can see on the first row that on chromosome 1, at position 187,141, we had a reference genotype of CGCT, and part of it was deleted, and the resulting genotype was only C. Now, you have how many reads we had for each genotype and the final genotype. Lastly, we provide a distribution of fusion genes in the genome. The circle is constituted by chromosomes. So each line in the circle represents a fusion gene. Red lines represent fusion genes in one chromosome, and the blue lines represent fusion genes in multiple chromosomes. The end of each line represents the fusion point. These are all most analyses that we offer in our standard pipeline. So now I'd like to further the depth of this talk into one of our advanced analysis workflow, the patient-derived xenografts, or we like to call it PDX. First, I like to start by defining what PDXs are. So as you can see in the picture, you have on the left the scheme of a human. This human was diagnosed with a type of cancer, and scientists took a sample of the cells of the cancer, probably through a biopsy, and implemented them in a humanized mouse. These cells can now be kept, multiplied, and studied in a more physiologically relevant environment when you compare it to a possible in vitro cell culture. So, to summarize, PDX models are used to create an environment that allows for a natural growth of cancer, its monitoring, and corresponding treatment evaluation of the original patient. So here in Novogene, we offer different workflows to answer your PDX questions. Due to the limited time that we have in this presentation, I will stick to the most complete and reliable workflow. If you are interested in knowing any of the other options, please contact us. So here's the workflow that I'm talking about, and I will guide you through it. On the left, we have the host raw data, which is in this case, the mouse. So you have already learned that here in Novogene, we use only clean data to perform any of our downstream bioinformatic analysis. So this will not be any different. So first, we clean the data obtained from the mouse, and then we map the clean reads from the mouse to the human reference. This is done so we can identify all mouse reads that overlap with human and remove them. Once we took the mouse reads that overlap with human, we can follow the normal RNA-seq workflow and obtain information of mRNA expression from the human tumor cells without worrying about possible contamination from mouse reads. So this methodology is particularly interested when analyzing the effects of treatment in patients. So for example, let's say that we have a cancer patient and we don't know which drug we can use to treat the type of cancer. Instead of testing drugs in the patient itself, which would lead to side effects and possible drug resistance problems, we can use an in vitro approach to test for the possible drugs. So following the scheme on the left, I'm showing you. So in the slide, we take some of the cancer cells from the patient and we introduce in a humanized mouse. These cancer cells will grow and we can propagate the cells in vitro. Using a common high-throughput drug screening test, we can select for possible candidate drugs or a combination of drugs. Once we have a few possible candidates in mind, we can test the effect of these drugs in the PDX model and move on with the drug that is strictly work both in vitro and in vivo. If one really wants to start understanding the molecular basis of how this drug works, a pretty good experiment is an mRNA-seq, which would give you a good idea of difference in gene expression and difference in biochemical pathways under the effects of the drug. This would for sure help elucidate the mechanism of action of this new drug. So now let's put everything that we learned in practice so far and see how scientists had actually used PDX and RNA-seq in order to review a new candidate drug for a nasopharyngeal carcinoma. So this is a paper published in 2018 on the Journal of Experimental and Clinical Cancer Research. The scientists in the paper established 5 nasopharyngeal carcinoma PDXs in a mouse model, and they applied potential drugs in 2 of them to assess their anti-cancer activities. RNA sequencing and transcriptome analysis were performed in 1 of them to correlate with the efficacy of the anti-cancer drug. So here on the left, we have the scheme of the experiment. They started on one side with the drug-treated PDX, and on the other side, as a control, the DMSO-treated PDX. RNA-seq and gene expression profile were performed, coupled with the differentially expressed gene analysis and the KEGG pathway analysis. QRT-PCR was then performed in order to validate the data. Here on figure 1, we have the differentially expressed genes on the panel on the left. On the right panel, we have the enriched KEGG cellular pathways. In this figure, we can see that there are 4 major pathways that are clearly being expressed differently, with the most evident one being involved in the cell cycle. Any cancer biologist knows that, that the cell cycle is extremely important in tumorigenesis, and the scientists then decided to further investigate this drug. On figure 2, you can see that the KEGG cell cycle pathway and the green boxes indicate downregulated genes in the cell cycle. This was specifically for the PDX model when it was treated with the drug POL, which stands for palbociclib. So here on figure 3, we have the validation of the findings. On the top panel, you have the RNA expression of 9 cell cycle related genes in the PDX system exposed to 5 drug treatments and compared to a control. So let me walk you through the top panel. Starting on the left, we have the control with DMSO, followed by the treatment with 5 different drugs. The last one is a combination of drugs, actually. On the panel in the middle, you can see the normalized number of gene expression of the cell cycle genes. If you take a look at the expression patterns of the cells treated with POL and GEM plus POL, you can see that most of the genes in the cell cycle are downregulated, while one of them are slightly upregulated. On the last panel in the bottom of the figure, you can see the validation through RT-qPCR. So, what was the conclusion that the scientists found? The drug palbociclib is a novel candidate drug in the treatment of nasopharyngeal carcinoma, and its effects rely on the downregulation of cell cycle genes. This is the reference in case you want to check the paper, and I want to thank everyone at Novogene who made this webinar possible, and also for you to taking the time to come join us. Thank you.
Awesome. Thank you very much, Mario. Before we move on to our live Q&A session, I just wanted to remind you guys of a few things. Again, thank you for participating. We will be sending out a link to this webinar that's being recorded, along with the slide decks that you saw here today, to the email that you guys registered for this webinar with. A few other things to note for future alerts on excuse me, upcoming webinars, promotions, and fun contests and activities that we have. We'd like to encourage you to follow us on LinkedIn, Facebook, Instagram, and Twitter. We constantly put out updates that are relevant to many of our services and would like to encourage you to take a look at that. We want to again thank you for joining us, and I'd like to ask if you have any questions, please do put them in the Q&A tool on Zoom, the Q&A module, I should say. We'll monitor that, and I'll give you guys a few more minutes just to let some questions come in before we can get those answers done. So with that, I'd like to hand it back over to Dr. Isidoro if you can answer a few of the questions that are coming in.
Hello, thank you, CB. So there are a couple questions here that I can see that are still open and I like to touch. So one is, what does mean Q2 end? So I think that the question that the client was trying to ask is, what is Q20 and Q30? So we here at Novogene, we promise a really high Q30 a possibility. So we offer at least, if your sample passes our quality control test, we guarantee that the Q30 of your samples is going to be above 80%. What does that mean? So the Q30 means that the probability of a base call be wrong is 1 in every 1,000, 10,000, 3 zeros. That's why the Q30. And so
Awesome.
the possibility of it to be wrong is 1 in 1,000. The next question that I would like to touch is what is the difference between total RNA sequencing and mRNA sequencing? So the main difference of mRNA sequencing and total RNA sequencing is how we select for the RNA that we're going to be sequencing. So in the case of the mRNA, we do a poly-A tail enrichment. So only messenger RNAs have the poly-A tail. So we're selecting mainly for mRNA sequencing. When we do the total sequencing RNA, we use a slightly different strategy in order to separate our input material. And in that pool, we're going to have mRNA, long non-coding RNAs, and all the regulatory RNAs that we have in the pool. If you are interested in that, please contact us. We're pretty good at doing that.
Awesome. Thank you.
And the last one that I would like to touch is I want to learn how to analyze my RNA-seq data. Is there a person that can teach me? So if you have any problems, we cannot particularly teach it, but we can for sure analyze the data for you. So even if you had not done your RNA sequencing with us, you can provide us the raw data and all the conditions of the sequencing run, and we can for sure analyze the data for you. But unfortunately, we cannot teach it.
Awesome. Thank you very much. And again, I'd like to remind everyone that they can find recordings of previous webinars and trainings that we've done on our website, along with other useful information. If you have a specific project that you want specific answers for, please feel free to reach out to us. We respond very quickly and we will provide you with customized solutions. Even if it's just to get some consultation, and we'd love to help you map out the different aspects of your project and make sure it's good to go. Thank you again for joining us. This is where we'll end this webinar. We will send out the recording and slide deck soon. Please look out for that. Thank you again for joining us. We appreciate your time. Have a great day.